series, and I want to extend our gratitude to all of you for making it a success so far, and also to our co-sponsors. We normally have some fancy slides up here thanking everyone, so I'll just name them. The co-sponsors are our department, Medical Ethics and Health Policy, ITMAT, the Office of Clinical Research, Abramson Cancer Center, and CHOP. And also, if you're inclined to tweet, if that's okay with you, Sarah, you can use the hashtag reps. Although, now I don't remember because it was on the side. It's like, do you remember off the top of your head? Okay. Penn reps. I think that's what it is. PENN reps. So others can follow along. And if you're new to reps, you can sign in our sign-in sheets outside the front door, and we'll make sure that you're added to all of our newsletters. And also, if you are interested in satisfying your human subjects protection training requirements, come see me afterwards and I can tell you a bit about a new online course that's available for free for affiliates from Penn and CHOP. We had a fancy slide about that too, and I can just tell you a little bit more about that course. Before I introduce today's speakers, I did just want to give you a quick preview of the rest of our spring lineup. Next month in February, we'll be joined by Dr. Karen Glantz, who will be talking on the ethical issues in the design and conduct of community-based research. And we're going to have 2 sessions in March. We'll have our regularly scheduled first Monday lecture, when we'll be joined by Dr. Allison Bateman-House of NYU, who will talk to us about right to try with commentary from Dr. Bob Vonderheide. And then you may have seen that we had to reschedule one of our fall sessions with Dr. Greg Gonzalez from Yale. He's a recent MacArthur Genius Award winner and really amazing person who has been a patient advocate and an epidemiologist, and he's going to talk about his work related to the role of patients in improving clinical research. So now on to today's talk. It's our pleasure to have Dr. Sarah Tishkoff here with us today. She's going to be presenting on ethical issues arising from her genomics research with indigenous populations in Africa. Dr. Tishkoff is the David and Lynn Silfen University Professor in Genetics and Biology here at Penn, where she holds appointments in the School of Medicine and the School of Arts and Sciences. She's a leading global expert in human evolutionary genetics, known particularly for her studies of African genomic diversity, which aim to better understand human evolution, adaptation, and disease. Dr. Tishkoff is a member of the National Academy of Sciences and a recipient of the NIH Pioneer Award, as well as other numerous accolades that if we went through, we wouldn't have time for your talk. And then after Dr. Tishkoff's presentation, we're going to hear some ethics commentary specifically on the ethics of some of what we've heard from Dr. Kellie Owens. Dr. Owens is a fellow in the ethical, legal, and social implications of genetics and genomics in our department, Medical Ethics and Health Policy. She received her PhD in sociology from Northwestern University in 2017, and she was a visiting fellow in the program on science, technology, and society at Harvard from 2015 to 2017. Her current work focuses on ethical issues related to the influx of genomic data in clinical medicine. And with that, I will turn things over to Sarah and keep my fingers crossed that the technology continues to work.
All right, see if this works. Can you hear me with this? Is that good? All right, now we're going to test this, make sure it works. It does, yay. Okay, so I am super excited to be here, to be invited to be part of this series of discussions. And I'm just going to give you an overview of the research that we do, which involves fieldwork in Africa. And then at the end, I'm going to talk about some of the issues that are specific to that kind of work that we do. It is, yeah. I think it's on, right? So let me just talk about what motivates our research first. So I believe there's an urgent need to do a better job at characterizing both genomic and phenotypic variation in ethnically diverse populations around the world. We're trying to understand evolutionary processes that generate and maintain that variation, including disease risk, and to understand how gene-gene, gene-protein, and gene-environment interactions contribute to both normal variation and disease susceptibility. The focus of my research is on Africa. And there's a number of reasons for that, but one of the main ones is that all modern humans originated in Africa. So these red dots represent the site of fossils of anatomically modern humans, the oldest of which is dated to about 300,000 years ago, was found in Morocco. After this African origin, somewhere around 50,000 to say 80,000 years ago, there was a migration of relatively small numbers of people out of Africa. When I say small, people estimate it was in the hundreds, maybe thousands, but not huge, and giving rise to populations around the rest of the globe. So you can imagine that this demographic history has shaped the pattern of variation that we see today. So keep that in mind when I show you some of the genetic results about patterns of variation. So this is from a paper perspective we published a short while ago, in which we looked at the genome-wide association study database. So, and we just looked at how many individuals in that database are of different ancestries, and showed that about almost 80% are European ancestry. About 10% East Asian ancestry, about 2% African, 1% Hispanic, less than 1% everybody else. And I think this is very problematic. This somehow actually got reversed. I meant to talk about this first. One of the reasons it's problematic has to do with health disparities that we observe. So for example, this is hypertension, which is more common in people of African ancestry than other ethnic groups. Type 2 diabetes, more common in people of Native American ancestry compared to other ethnic groups. And often these are comorbid together. Now, there's no doubt that there are strong environmental, social behavior factors that are contributing to these differences, but it's thought that there are also differences in genetic risk factors as well. And yet, just showing results from another database, NOMAD is a commonly used database that includes sequencing data, exome sequencing, and some whole genome sequencing data. And again, predominantly European, people of European ancestry in that database. Some of you might have heard about GTEx, again, predominantly European ancestry. So I think this is problematic for a number of reasons. One is that it impedes our ability to fully understand the genetic risk factors for human disease, and I think it can exacerbate health inequalities. Our ability to translate genetic research into clinical practice or public health policy could be incomplete, or even worse, it could be mistaken. I don't know if you may have heard about polygenic risk scores, which basically are derived from these genome-wide association studies. You look for an association between genetic variants and a disease trait. And if you just add up all the associations, you're looking at genetic markers across the genome, add them all up, and that tells you something about someone's risk for certain diseases. But what a number of studies have shown is that when these are done in Europeans, and they all have been pretty much to date, that they don't work across other ethnic groups. They're essentially completely uninformative. And people are applying this right now in many clinics to date. And also, one of the ways that people determine if a variant is pathogenic is they'll go to some of these databases and they'll ask the question, do I see this variant? Is it common? Because if it's common, it's not pathogenic. So if they see that it's not common, they might think it is pathogenic, but it could be that it's in other ethnic groups other than Europeans, and we just don't know about it. So in order to help eliminate this disparity, together with a number of African collaborators who I've highlighted here from University of Botswana, Cameroon, Kenya, University of Yaoundé Medical School in Cameroon, University of Khartoum, and Addis Ababa University. We've been doing fieldwork in Africa for the past 19 years or so. So I wanted to show you a bit of what that's like, and then we'll discuss some of the issues that arise from this research. So this is from Ethiopia. And I like this picture because it gives you the outside perspective of the car. We have to bring all of our supplies with us. We mainly focus on studying populations that practice indigenous lifestyles. They tend to be in very remote regions. And this gives you the inside perspective, which is basically super crowded. And this is from Tanzania. These are two hunter-gatherer groups that we studied called the Hadza. This is sort of the setup for collecting anthropometric type of data. This is me returning results, but I'll get back to that later. These are the team, our collaborators from the University of Yaoundé Medical School, and some of the people who helped lead this research, including Alessia Ranchero, Eric Mabunwe, who was a postdoc in the lab, and Megan Rubel. Now, when we do this research, we run into a lot of challenging conditions. So one is what happens to the roads in the wet seasons. And secondly, in Cameroon, these giant logging trucks, because most of the time we're on those other type of roads, not this type of road. And if it's in front of you, it's super, super slow and it's kicking up dust and you can't see to pass it. But when they're behind you, they're going like 90 miles an hour and running you off the road. So just one of the risks we face. But throughout this research, from the very start, we were really careful to consider the ethical issues around doing this type of research, which means not just going through IRB review here. We have to go through very detailed ethical review in each of the countries that we work in. That takes a long time. I would say on average, it's about 5 years before we actually start doing field work. From the start when we set up collaborations, go through extensive rounds of ethical review, and then we also go to meet with the community. So in Cameroon, for example, we spent an entire year before we initiated research just going to all the communities, telling them about the research, why are we doing it, what are the potential risks, potential benefits, if any, and so on. And in this particular group, when we went back, they were kind of annoyed because they thought we were finally going to do the research, but that was a bit later. The other thing that I think is really important is to actually return results to individuals. This is the Hadza group that I mentioned. They don't typically read science, but what we do is we do a layman's summary and then translate it into the local language. And we can also literally just show them some figures. I'm going to actually show you some of them today. And I found personally that they were extremely appreciative and particularly interested. One thing you might wonder is, are they interested, how do they feel about getting information about ancestry or their population history? In Africa, the groups that we interacted with really, really appreciated that information. So it shows you there are cultural differences, because in Native American groups, they may not appreciate that, or in Australian Aborigine populations. The other thing that's really important is training and capacity building. And so these are some of the grad students and postdocs that I've trained in my lab. Just to show you a bit of what we actually do in the field. So we're collecting samples, DNA and RNA and frozen plasma from blood. We get detailed ethnographic information, information about diet and any medical history that they might have. And then we have to process these samples in areas where there's usually no electricity. We used to hook up the centrifuge to the car battery, but now what we do, it's off the screen here, but we basically take a generator with us and use that, just hook it up, and we can set up a lab in the bush. We're also collecting information about variable traits. So very detailed anthropometric traits like height and weight and limb length, limb circumference, grip strength, skin pigmentation, a number of cardiovascular, lung, and blood phenotypes like blood pressure and hemoglobin levels. We look at metabolic function, lactose tolerance, glucose tolerance, and infectious disease status when we can. And then what we're doing in my lab is we're doing an integrative, using different types of omics data, so characterizing whole genome sequence data, gene expression, microbiome, gut microbiome, the metabolome from the blood. And we integrate that together with information about the environment to understand how these are contributing to normal variable traits and sometimes disease risk. Now, when studying African populations, these particular populations, as you're going to see, they're genetically very diverse within and between groups, and their environment's very different between groups. But within any particular population, the environment's extremely homogeneous because these are really remote rural populations. They tend to eat the same thing. They tend to either very limited amount of healthcare. They're going to tend not to be on any pharmaceuticals. And so you're able to control for the impact of environment and genetics on these variable traits. But one thing you could do is control for ancestry, look at people of similar ancestries that are living, for example, in a rural versus an urban environment to try to distinguish environmental from genetic factors influencing variable traits and disease risk. Or you could do the opposite. These different colors represent inferred genetic ancestry. And these are two groups that live next to each other in Cameroon. We could see they have very different genetic ancestries. The Fulani is a group that we're very interested in studying, and one of the reasons is that they have a relative resistance to malaria for reasons people don't know. It has nothing to do with sickle cell and the typical known genetic variants that provide protection. The second thing is they have an unusually high incidence of diabetes and hypertension. And in fact, we hypothesize those might be related. There might be some altered immune response that's adaptive in terms of pathogens, but it may put them at risk for hypertension and diabetes. And then the group that lives nearby, they're in the same environment, but they don't have the high incidence of diabetes and hypertension, for example. And then in southern Africa, the San hunter-gatherers and the neighboring Bantu agriculturalists, very different ancestries. The San have a high predisposition towards TB infection for reasons we don't entirely understand. So I want to tell you a bit about patterns of genetic diversity. This is from a study we published a number of years ago, but it remains one of the largest studies of diversity in Africa. We looked at highly variable short tandem repeats in over 3,000 individuals from 120 or so different populations, mainly in the places where we and our collaborators are doing field work. And you can see there's still quite a bit of gaps there. So the first thing is that we found, as everybody has so far, that the levels of genetic diversity are highest in Africa compared to other regions. And you see a decrease in diversity as you go west to east across Eurasia into East Asia, Oceania, and the Americas. You can also use computational methods to infer individual ancestry. So these different colors represent genetic ancestries. The lines are people, and people can have ancestry from these different populations. So here are some of the non-African populations. In blue are people who self-identify as European or Middle Eastern. People who self-identify who are from East Asia, Central Asia, India, Pakistan, and this was Oceania and the Americas. And we could see in Africa here that there's quite a bit of colors representing all the genetic diversity that exists in Africa, which is extensive when you compare it to the rest of the world. And if we zoom in and just look in Africa, and then I just took those lines and I pulled them together basically by region and showed the percent ancestry in different regions, you could see a lot of variation in Africa. So people from Eastern Africa have differing ancestries from Western and Southern and Northern Central. And then the other thing you see is the mixed colors, right? So lots of admixture. The history of Africa is migration, subdivision, coming together, intermarrying, and so on. So you see a lot of variation. This is from a recent study that we haven't published yet. It's of high coverage genome sequencing from 180 individuals from 12 ethnic groups in Africa, representing some of the most of the diversity, those different colors that I showed you. And the point I want to make here is that by looking at a relatively small number of people, we found millions of novel variants, almost 6 million that are not in any current database. Many of them are predicted to be functional. And that's what you find just by looking at a relatively small number of people representing all the diversity in Africa. We can also make a phylogenetic tree from this data. And based on genetic distances, we can see that the populations that have the oldest genetic lineages are the two San populations, Junwasi and Tzu. If I could speak with a click, I would, but I can't. So these are one of the few click-speaking populations in the world, followed by the Baka Pygmies from Central Africa, other African groups, Amhara from Ethiopia, and then the non-African populations, which have essentially a subset of the diversity seen in Africa, and that makes sense based on the demographic history that I showed you. Now, the other thing that my lab studies is adaptation to different environments. And this is from a paper we published in Science, giving examples of local adaptation. I think many of you are familiar with the sickle cell case. That sickle cell mutation is common in Africa because people with one normal copy and one sickle cell copy have protection from malaria. We've also looked at things like lactose tolerance, which is thought to be an adaptive trait in populations that practice dairying cattle domestication. So to give you an idea of how we collect this type of data, for this we do something called a lactose tolerance test where people drink the sugar lactose found in milk. We then, every 20 minutes, follow up with a test of blood sugar using this diabetes monitor. And then we look at the maximum rise in blood sugar. And people who are above 1.7 millimolar glucose rise are considered to be lactose tolerant, and these individuals are lactose intolerant. But we actually studied that as a quantitative trait. And then we sequenced a candidate region that had been identified in Europeans, but we didn't see the European lactose tolerance mutation in our data set. Instead, we found 3 novel variants associated with lactose tolerance that are regulating expression of the gene lactase. They're very close to the European variant, but they're very restricted. So this variant at position 1410 is really pretty restricted to Tanzania and Kenya, and then places where people migrated from that region, so into southern Africa, for example. This variant is common in the Middle East. It was introduced by migration into North Africa, and this variant appears to have arisen in the Horn of Africa. And then we look for signatures of selection. So people who have 2 copies of the variant associated with lactose tolerant, shown in red here, if you look at genetic markers going out 3 million nucleotides from that region, you can see that this homozygosity is extended. And that's because this was such a beneficial variant that it rapidly rose to high frequency and it dragged with it all the neighboring variation. And if you look at the chromosomes that have the ancestral variant, so this would be intolerant. So lactose intolerance is the ancestral state. So it's only those people who have this adaptation to cattle domestication and dairying that have this variant. But if you look here, you don't see that at all. So that's a very striking signature of selection. The other adaptive trait we're interested in is skin color. So this is showing melanin levels. Melanin is the dark pigment in skin. And we could see that the levels are strongly correlated with UV exposure. So it's thought that this is an adaptive trait, that when modern humans left Africa and went to higher latitudes, that there was selection for lighter skin, because it's thought that this has to do with the fact that vitamin D is synthesized in the skin in response to UV exposure. So if you have darker skin, you're not going to produce as much vitamin D, you have less UV, so the thought is you need to have lighter skin to promote more production of vitamin D. At lower latitudes, there'd be selection for darker skin. It's protective against cancer and folate degradation, for example. But as usual, almost all studies were in Europeans. So to look at this in Africans, we used called a spectrophotometer, where you basically shine a light under the arm and you look at the reflection, and based on the wavelength of the reflection, you can infer the skin pigmentation levels. We see a huge amount of variation in Africa, and I want to point out that this is not due to admixture with non-Africans. This is naturally occurring variation in Africa that people just didn't realize how much variation there is. So the most lightly pigmented people are the San. Remember I told you the San have the oldest genetic lineages in the world. We see a lot of variation. The most darkly pigmented people are those from South Sudan. They originated in South Sudan. They speak Nilotic languages. They practice cattle domestication. So they have the darkest skin color in the world. We then did a genome-wide association study looking at genetic markers across the genome in association with skin color, and we found a number of novel loci, variants associated with skin color. I'm not going to get into all of them today, but I will just point out that one of them was a gene never previously identified. If you go to PubMed, this is actually the first paper to describe it. When we knock that gene out in a mouse, it has a really dramatic impact on pigmentation. It's now been shown that this gene plays a role in risk for melanoma as well, so there are medical implications. And if you look at the frequency of the variant here, this is in a regulatory region. It regulates expression of this gene. The variant associated with light skin color is common in Europe and East Asia, and also in India, Pakistan, and Eastern Africa, and in the San. The variant associated with darker skin color is common in Africa, South Asia, and Australo-Melanesia. And these are populations like Afro-Caribbeans of recent African ancestry. So what we found is that we found 8 novel associations, and they're not all showing up here for some reason. This is off of Mac, so there must be some translational issues. But in any case, in half the cases, we found that the ancestral allele is the light-associated allele, not the dark-associated allele. And furthermore, if you look at the age of the derived mutations, they're almost all preceding the origin of modern humans. So the point is, there are light and dark variants segregating in Africa for up to 1 million years. Now this poor chimp lost its hair because it has like an autoimmune disorder, so it lost all of the body hair. But you can see that the skin is relatively lightly pigmented. Anthropologists think that this may have been the ancestral state, is to have lighter colored skin. There's no need for them to have darker skin if they have this body hair. But when our ancestors left the savanna, or left the forest and went into the savanna, there'd be selection to lose the body hair, increase the number of sweat glands for thermoregulation, and then if that's the case, there would be selection for darker skin. But I would point out that we know that both dark and light variants have been segregating. So we don't know exactly how darkly pigmented these populations were, and there could have been variation in Africa itself. The other interesting thing, I'm just going to check how I'm doing on time. Better hurry. The other interesting thing we found is that some of these variants associated with dark skin are also common in South Asia and Australasia, other places where we see darkly pigmented skin. In this case, they were introduced during that out of Africa migration. All right, I'm going to spend about 5 or 7 minutes or so on this last part. This is actually from a commentary that we published with Mike Yudell and Dorothy Roberts and Rob DeSalle, talking about the need to take race out of human genetic studies. And that's because, particularly in the medical literature, you'll see people using these terms that often infer a biological connotation of race or definition. My biggest pet peeve is Caucasian. I hate that word. We should just get rid of it from the medical literature. So how do you classify race? This is something that I presented this in high schools, for example, and undergraduate classes, and I asked people, how do you classify race? First thing they say is skin color. It's really a terrible classifier of race. We see people of dark pigmentation in many different regions of the world. It's an adaptation. And as I've shown you, it turns out that many of the variants that are playing a role in light skin color in Europeans, they actually originated in Africa, is what our study showed. The other problem is that, of course, biological classification of races has been associated with hierarchical ranking of races, biological determinism, eugenics, justification of genocide, for example, the Holocaust, colonialism, slavery, and a number of other really bad things. And part of the problem has been that I have found when we've had discussions about this, is there's always this sort of intertwining of sociocultural and biological classifiers, which makes it nearly impossible to come up with a really solid definition of race. Certainly based on what I've shown you, race does not correlate with the patterns of variation, right? I showed you that there was more variation amongst different Africans than many other places of the world. How could there be an African race, right? We're seeing more variation between people in different regions. We do know that people differ for risk for disease, and people whose ancestors lived in different environments have been subject to different selection forces and have different demographic histories. Some diseases may be influenced by geographically restricted alleles, and information about individual ancestry could provide important medical information for diagnosis and treatment. But we have to be aware of racial profiling. So, for example, somebody who has anemia, and they might be of African ancestry, but it turns out they have grandparents from the Mediterranean, and it turns out it's not sickle cell, it's another type of variant that could be causing the disease. And also, we can't make generalizations based on continental region of origin because there can be so much variation within even a region. And then, of course, we have to take into account environmental and social behavioral factors as well. I'm going to skip over this and just go to some of the last slides, which is to show you what we did. I was part of a workshop that took place in a place called Sumkwe in Namibia. This was organized by an anthropologist, but there were legal scholars there were sociologists, there were policy people there. And we wanted to get the feedback of the indigenous groups, the San populations, about genetics research to inform them what people are doing with their data, because they're one of the most, how would I say, they've been studied quite a bit they're of considerable interest from geneticists because they have these old lineages. but nobody's rarely gone back and told them what people are doing with their data. So one thing we did on the way there, we went through Botswana, we returned our results to people along the way, and had very good discussions. I can talk about that later if it comes up. Then we went to Tsumkwe in Namibia, and this was how we did our slide presentation, which is put up a sheet and tape it. And we had two groups there. So they represented, oh, it's not coming out really well here. I don't know if there's a way to dim the light or not, but there were basically two groups of the San. One of them is from that region, Tsumkwe, which is, how do you say it? It's I forget the word for it, not conservation, but they basically have a very strong community structure where they benefit from things like they sell jewelry and art, and all that money gets pooled and goes to a common source. And they're able to maintain some of their traditions as well. Now the other group is from an area where they're basically squatters on people's farms, okay? So it's a totally different situation for them. And if this works, I don't know. Let's see if this is gonna work. This is what caused us so much trouble before. All right. Here it is. All right, now how do I escape from this? Help me escape. Just hit it again? And then I can just go forward. Okay. So what that was, I don't know if you guys could hear it. It's actually interesting because they speak with clicks. I just think they have a beautiful language. But every time we would tell them about the research we were doing, not just us, actually most of the research wasn't our own, it was other geneticists. And we started with what is a cell, what is DNA? And we had to do this for people who don't have any formal education, most of them. But I think we were successfully able to do that. We did it over 2 days. And then we told them about the research and what the results were. And then at the very end, we said, well, what are some of the issues and concerns that you have? And this is what they came up with. Who benefit? Should it be individual versus collective? And remember, I told you the two groups. So when we asked them what they wanted to get from this, that was the most heated discussion that I saw. One group went outside. They wanted to talk about it privately from the other group. But the ones who had the collective, well, one of them wanted, you know, those individual colors, the results that show the different ancestries, like 23andMe, they wanted those results. They literally wanted to know their ancestries, like 23andMe. But we pointed out that's a problem because if we give back individual results, the other issue was paternity, that we would find out non-paternity. So we explained why we thought this would be a problem. They said, okay, we agree. That's a problem. We're not going to expect to get the individual results back. Payment, so we typically don't pay because it can be considered coercive, but then, you know, we give a small gift. What should you give? One of the groups wanted something to go to the community. So give some amount per person that goes to the community. We'll distribute it as we want. The other group is like, no, we just want cash. They're living on farms. They don't have this community organization. They were concerned about intellectual property, engagement, confidentiality. I don't remember what differentiating was. How to deal with bad researchers, so people who are not using ethical practices. Publications, should they have a role? Should they be able to say what goes in publications? I think that raises a number of concerns as well. And then I forget what GRN stood for. So I'm just going to end by, I'm just going to leave it up there, and I think you can take it from there. I will say, however, oh, I forgot. I do want to mention we have a new Center for Global Genomics and Health Equity. Our goal is to promote health equity through global genomics research, education, policy, and practice. And so we're going to be doing this through seminars and through pilot funds. And I'm happy to say we just got 2 fellowships, postdoc fellowships, that are going to be the Penn Health Equity Fellowships. They're for people who represent groups that are underrepresented in the sciences, and they have to study health inequalities. So if you're interested, you have some candidates, let me know. And now we'll go back to the ethical issues, and I will let Kellie take over.
Seems like it's on.
mouse is over here. I see why that was a problem.
It shows up here, I think. You just go to slideshow. Trying to figure out how to get the mouse onto the other screen.
Literally disconnected.
I went, hold on, that's not today.
Oh, thank you. Okay. Yes, perfect. Did it. All right, thank you all for coming. I think my role as a commentator is just to ask some additional ethical questions about genomics research. Perhaps Sarah can help us think about them, or the larger group. We can talk about them together. There is, of course, already a giant literature on ethical issues in genomics, a bunch of which is focused on ethics related to studying populations of color or of disadvantaged communities. There's no way that I could talk about all of that research. So as I was trying to come up with what I wanted to focus on today, I decided to sort of play to my own strengths, which is to talk about some of the ethical issues that are most salient for social scientists like myself. I'm a sociologist by training, and some of the things that we think should be better represented in the ethics world when it comes to genomics research. So those types of issues tend to be larger scale. So Sarah talked about a lot of ethical issues related to the implementation of genomics research. I'm instead going to try to focus on higher level ethical issues that we would solve more on the level of institutions or organizations. So I'll start, this is a slide from Dr. Tishkoff's talk with perhaps what's the most obvious ethical issue, as she's already mentioned, with current genomics research, that there are just way too many people in our samples of European ancestry when compared to other populations. Dr. Tishkoff talked about that already, about why scientifically we need to include more diversity in our samples, but I think that inclusion of other populations isn't just important scientifically, it's also quite important politically. And here I'm going to build on work from a sociologist, Stephen Epstein. He wrote a book called Inclusion, The Politics of Difference in Medical Research. And he traces the emergence of this political movement, suggesting that inclusion of women and people of color was important in medical research. And it started about in the 1980s, driven mostly from women's health groups in the United States. And the idea originally was that historically disadvantaged groups were being left out of research and that their health needs were then compromised by that. So by including more diverse groups, we could then start to address health problems that might differentially affect those populations. But he also talks about what I'd call the paradox of inclusion that Dr. Tishkoff has already started to mention. Which is that by focusing on differences between groups and the importance of enrolling different types of participants from different social and ancestral backgrounds, we also have a potential danger of the reification of the belief that the differences between those groups are essentially biological rather than social, economic, or political. And we've already heard a bit about the history of eugenics and other atrocities. That makes genomics researchers and social scientists incredibly attuned to social dangers of promoting ideas about biologically based differences between social categories like races. So I think by emphasizing the importance of including diverse groups, people like Stephen Epstein would argue that then we have failed to demand adequate attention to the crucial set of issues, which specifically are the ways in which inequalities and power differentials in the broader society affect people's exposure to health risks, their capacity to access quality medical care, and the likelihood that they'll be subject to conscious or unconscious discriminatory treatment. Which, again, is not to say that we shouldn't be diversifying the types of populations in genomic research. Of course we should, but also that it's incredibly important that we make sure to understand how this research is being brought into the public sphere to try to reduce the likelihood that there will be discriminatory effects of research on population differences. And I think we have a responsibility to help shape those conversations that end up being about race. I'll just say quickly for the sake of time that I think it's even harder for us to figure out how to have these conversations in public spaces because even the experts can't really agree on how to talk about race and genomics. You may have seen this come out about 2 years ago now, an op-ed from David Reich, how genetics is changing our understandings of race, and very quickly a response from a bunch of prominent social scientists and others who critiqued his understanding of race by saying how not to talk about race and genetics. I think for the sake of getting to questions, I won't go too much into this, except that the whole point of his original op-ed was to say that he was worried about whatever discoveries are made, that they'll be cited as scientific proof that racial prejudices and agendas have been correct all along. And he says, as a geneticist, he knows that it's simply no longer possible to ignore average genetic differences among races, and wanted to develop a candid and scientifically up-to-date way of discussing any of those differences. Social scientists, including myself, would disagree with that characterization. Again, not disagreeing with findings from the work like Dr. Tishkoff's, but saying that there's also a robust body of scholarship recognizing the existence of geographically based genetic variation in our species, but that variation is not consistent, as we've already heard, with biological definitions of race, nor does that variation map precisely onto changing social definitions of racial groups. So that these social scientists would argue there's a difference between finding genetic differences between individuals and constructing those genetic differences across groups by making conscious choices by humans about which types of groups matter. So I would just say then that again, we have this responsibility in doing research on differences between populations to control or at least shape the public messaging surrounding that, but that can be extremely difficult if we can't agree in ourselves. So I think what that means is we need to have a lot more conversations about how to talk about race and genetics. The second and final point I want to make is related to justice in ethics of genomics. Bioethicists, I think for better or worse, tend to focus on three principles in bioethics: autonomy, beneficence, and justice. And generally, a lot of the research on ethics in genomics has been focused on those first two of the three principles. We have a lot of good research, a lot of good practices, some of which you've seen Dr. Tishkoff talk about how to do things like good informed consent, good benefit sharing, good community outreach, those sorts of things. But we are less good at creating a framework to handle the principle of justice as it relates to genomic science. There are people trying to do this. So here I'm drawing mostly on the work from Jenny Reardon. She wrote a book called The Post-Genomic Condition: Ethics, Justice, and Knowledge After the Genome. And by post-genomic condition, she means now that we have access to genomic technology and data, what do we do with it? What is its meaning and purpose in a social sense? She applauds genomics researchers for making their research more democratic by making it anti-racist in a lot of ways, but also agrees that we need a better apparatus to handle justice issues that rise above the level of individual research projects. So she asks, for example, what is the relationship between innovation in science and technology and inequality? The growing gap between those who have and those who have not is perhaps the defining issue of the first decades of the 21st century. It also arose time and again as people gathered over the decades to make sense of the meaning of genomics in their lives. What sense does genomic research make when basic healthcare is beyond your reach? This question deserves our collective attention. And I think this is a big question that can't really be answered by one researcher or even one institution. So Reardon gives the example of a project in Nigeria where the community members asked for a community health center in exchange for participation in genomics research. And at first, institutions like the National Human Genome Research Institute weren't really sure how to respond to that. In the end, they did end up supporting the expansion of hours for the community health center. But they also decided that it wasn't really in their purview to support new medical centers. They are a research institute. But I think that deflects the responsibility to consider larger justice issues. So whose responsibility is it then, if it's not NHGRI's, if it's not individual researchers, to ensure that communities can have real access to the medical care and benefits generated from the research that they participate in, or even more fundamentally basic medical care at all? I think if NHGRI isn't equipped to handle those issues, who is? I think we need to start having these bigger conversations, even if they're incredibly hard to answer, and to come up with better frameworks to address how, at the one hand, we have great riches that come from genomic research, but also deep deprivation of many research subjects to be able to access healthy environments. So I'll just stop by asking a few questions and provocations, either for Dr. Tishkoff or the group. First, what responsibility do genomics researchers have to the public uptake of their research on differences between populations? And can we agree on how to discuss those differences, especially when it comes to race? What responsibilities do we have to the African communities participating in genomics research? And how can our frameworks for benefit sharing in research address growing social and economic inequality? Thanks.
So I will walk around with a microphone just because we are recording this. If you have questions for either or Dr. Owens, start with one. So you had mentioned it takes about 5 years to get a project off the ground. And so do you mean from when the project is first conceptualized or when the funding starts? And I'm interested to hear whether you find that 5 years worth of work and ethics review and community consultation valuable, or whether it's kind of a bureaucratic slog, or both. Like,
It's both. Yeah, it's both. No, when I say from start to finish, I mean in terms of you have to, of course, identify partners. That's having partnerships is critical. And I'll be perfectly honest, depending on the country, this can be extremely challenging. So in fact, I was recently interested in maybe doing research in Namibia, and I've been trying to identify geneticists. I can't. I mean, I think there might be one in the country who has done some genetics. That's what we're talking about. I mean, so you can have the best intent. There could literally be a handful of people who had that expertise. And in one of the areas that we did research, that one individual who is a geneticist is completely overburdened with other responsibilities that he has at the university. So you can have very good intent, but it can be challenging for some practical reasons to find people who are equal partners in terms of the academic endeavors, I guess you could say. But the first part is try to find them. We have to talk about what are their interests and what can we do. Benefit sharing is always something that comes up. What are we going to do to help train students? Sometimes we provide equipment for laboratories, training so that they can conduct the research themselves. And then we start going through the ethical review, and these, honestly, in most of the places, they didn't have major ethical concerns. It really was the bureaucracy. It was, they only meet every 3 months. Somebody didn't show up to the meeting, cancel it, start all over again for the next 3 months. They didn't like the title, start all over again for the next 3 months. I mean, this can just go on and on. There were some valid ethical issues raised in certain places, and we had to be really, really careful. It took a long time of discussion and going back and forth, and me having in-person meetings with the ethics review committees to explain, when they had questions about things, that there's no substitute for being there. So that's what took so long, and yes, it is frustrating sometimes. So the longest one was 9 years, but that one was because, again, political reasons. It was in Ethiopia, and it turns out we were probably being blocked by someone who was like working with a competitor, and then, as happens in Ethiopia when there's a change in who's in charge, things tend to change rapidly, and that's when we were able to get this permit through.
We have a question.
Thank you. Thank you, both of you. The talks were amazing. My question is in reference to informing researchers and clinicians in the United States based on the information that you're gathering in Africa. So for instance, you said the Fulani have a higher prevalence of hypertension and diabetes, which is always assumed in the United States that people of African descent have a higher prevalence of that. You're not finding that in other groups in Africa. So how is that information being used to inform clinicians and scientists in the States?
Thank you for that question. So I just got an award from the American Diabetes Association called a Visionary Award, and it's for people who don't study diabetes like me. And so it's trying to get different perspectives. And it was focused on this question about the Fulani. And one of the things, one of the reasons that they wanted to support it is, one, it's just that there has been so little research done on people in different ethnic groups in Africa. But the other thing is, from that study I showed from Science, the one where I showed all the different colors and everything, I didn't show the results, but we actually looked at African Americans as well from 4 regions of the US, and we looked at ancestry from different parts of Africa. Not surprisingly, almost all the African is from the western parts due to the history of the slave trade. But that Fulani group is actually, they go from the Sudan all the way across sort of central Western Africa, and their language is actually a Western Bantu-related language. And so we've shown that African Americans can have up to 14% Fulani ancestry. This type of diabetes, I'm not making any claim that this explains the high prevalence of diabetes in African American community has very, very strong socioeconomic behavioral factors that are playing a role. But this type of diabetes was a little different because it is not correlated with being overweight. And so their BMI on average is about 19. And despite having a very low BMI, no obvious risk factors, they're getting this diabetes. And there's a subset of people in the US as well who are the same way, that they don't have these obvious risk factors, but they're getting diabetes. And that might shed light on that.
So my question actually picks up on that last one in a way. Currently, when we ask about race for medical purposes, we tend to lump together, African American, European American, Asian American, whatever. Which you appropriately critiqued. Question is, how do you envision using this more refined sort of genetic ancestry to incorporate that into medical care? Which I think is maybe another way of saying the question that was asked just a moment ago.
So I think, one way to start is to actually ask individuals about ancestry rather than classifying by race, because somebody who might be classified as African American, particularly in the Philadelphia area, we have over 50,000 immigrants from Africa, and they could be from Eastern Africa, and I've shown you that people from Ethiopia have very different genetic variation than, somewhat different, not very, that's relative, but from people from West Africa or Central. And so I think getting as much information about ancestry as possible, and then the ultimate goal in the future is going to be that people are turning more and more towards genomic analysis at the individual level, because ultimately you want to know for that particular person, if there's, say, a gene that is playing a role in drug metabolizing, you want to know that particular region, what are the variants that that person has. So then we're getting down to very personalized medicine or precision medicine tailored to the individual. That's the ultimate goal, but we're just not quite there yet.
Can I ask another question about the funding of your work, right? So how is it that you can get this kind of work funded? Who's funding it? And if it has such a long trajectory from start to finish, and there are all of these ancillary issues about benefit sharing and paying for things that are not necessarily traditionally part of research, maybe you could just talk a bit more about how the finances work.
Well, maybe I should start by saying that it's super expensive. So each field season is typically, well, in Cameroon, for example, I had Eric Mabunwe, who was a postdoc in my lab, spent the year before. He was there for a year prior to starting the actual research, going to all the communities and things like that. But once we actually get in the field, it's about 6 to 9 months in the field to collect the data. And we have to, for example, I showed you that car, right, to get to those remote areas. So that car would cost about $200 a day for rental, not counting paying the driver. Add that all up over 6 to 9 months, it's about $40,000. So I could buy a car, which I actually ended up doing in Cameroon because it was more cost effective. So in total, it's about a good $200,000 per season, at least, to do this research. And mainly I paid for this through career awards. And that's what the benefit was of having these career awards, where we could have the ability to do more creative research, do different things with the funds. It gets increasingly more challenging to do this, I have to say, because NIH, they have funded some. So NIGMS has funded some of our research. NSF definitely has a history of funding field work, but it does become quite challenging.
So I was very happy to hear that you do go back and disseminate information about your research. I'm wondering what infrastructure might be developed as you're setting up temporary to do the research that you're doing at the time. But is there any internal or infrastructure being built for long-term for the people who live in those areas?
Infrastructure for research or for other aspects of healthcare? For research. That's a goal of mine. I think that would be really important. Now, Penn has this Botswana-UPenn partnership, right? And I have done some research in Botswana, and that's actually been very helpful because it's also a way that there's benefit sharing, that there's training of students and helping to build the hospital there, the medical school, so on. But in the areas that I work, it's extremely challenging from a logistical perspective. Just in terms of having the stability, people in extremely remote areas, the lack of infrastructure, the lack of electricity, the political instability that can just, everything disappears if there's a new elected leader. So we're interested in that, but we're going to have to think of what's the right place. So one possibility that I think is a very more relatively stable would be in Tanzania, in the Arusha area where we do a lot of our research. That might be one possibility that we've thought about, but I think it would be wonderful if we could do that.
Okay, well, I think we're out of time, so thank you both for a really compelling discussion.
Good job. Short notice.